The overall goal of this procedure is to demonstrate how to culture and record electrophysiological activity of auditory neurons on multi-electrode arrays.
This method can help answer key questions in the hearing research field, such as characterizing the electrophysiological properties of auditory neurons and their stimulation by the electrode arrays. The main advantage of this technique is that it allows for extracellular, non-invasive, simultaneous recording of the neuronal activity of large number of auditory neurons.
The implications of this technique extend towards therapy. In fact, they could be used to implement the electron-neuron interface of neuroprosthetics like cochlear implant to restore hearing in deaf patients.
To begin this procedure, prepare the ECM coating solution by first thawing the ECM mix on ice. Then, dilute the ECM mix in basic culture medium at a ratio of 1:10 and store it on ice. For new MEAs in a laminar flow hood, immerse them in 70% ethanol for 30 seconds. Subsequently, wash them with distilled water for 30 seconds. Then, let the MEAs dry for 30 minutes. To coat the MEAs, pipette 50 microliters of coating solution over the MEAs with a cold 200 microliter pipette tip. Then, allow the MEAs to coat for 30 minutes to 1 hour at room temperature. After that, remove the coating solution, add 100 microliters of culture medium supplemented with 10% FBS and 5 nanograms per milliliter BDNF, and leave it at room temperature until plating the tissue. Next,